As we all know, these miniaturized three-dimensional structures mimic the cellular architecture and functionality of human organs, offering unprecedented insights into these disease mechanisms and, of course, potential therapeutic strategies. So in today's session, we will delve into the use of preformed fibrils of tau and alpha-synuclein proteins in modeling neurodegenerative diseases. We will also discuss the application of chemicals such as aftin-4 to induce pathological features of Alzheimer's in cerebral organoids. Whether you are brand new to the field of organoids or an experienced researcher, today's webinar promises to provide valuable perspectives and foster discussions that could pave the future, the way to the future, and to breakthroughs in disease modeling and drug development. Most importantly, I'm delighted to introduce our speaker for today's presentation, Idil Ariöz, our field application scientist here at ACROBiosystems, specializing in neuroscience. Idil has an extensive background in neuroscience research, having conducted academic studies focused on neural regeneration, spatial distribution of synaptic proteins, and molecular targets in epileptogenic neurodevelopmental diseases. Today, she will be our host and guide us through the aforementioned discussions. And please, we encourage you to engage with Idil and ask us questions throughout the session to maximize this opportunity for discussion. And yeah, once again, thank you all so much for joining us, and let us look forward to an engaging and informative session. Idil, please take it away.
Thank you very much, Mary, for the nice introduction. So my name is Idil Ariöz. I'm the field application scientist at ACROBiosystems, and today's webinar, we will talk about advancing drug development and disease modeling with human iPSC-derived organoids. So let me introduce ACROBiosystems to you. At ACROBiosystems, we offer life science tools and solutions for applications ranging from drug discovery, research, and commercialization. Our catalog contains over 4,000 recombinant proteins to suit your needs. We've also expanded into organoid solutions to advance drug development and disease modeling. We were founded in 2010. We have grown over 800 employees across the entire globe, and our locations include Europe, North America, and the APAC region. We serve over 9,000 end users across the globe, and we have collaborated closely with the top 20 pharmaceutical companies. We're excited to help provide innovative solutions to customers in over 70 countries and for more than 50,000 scientists. Our products include recombinant proteins, antibodies, assays and kits, GMP-grade manufacturing, innovation, lab services, as well as organoid solutions, which I will talk about more in detail today. We have unique product platforms to help your needs, for services such as complex structure protein development, AI-based protein design and optimization, scaling up protein productions, protein labeling conjugation, protein characterization platform, and immunoassay developments. You can reach to us. Here is an overview of core products and services in the space of recombinant proteins, antibodies, and enzymes. So let's start with organoids. Organoid definition is a three-dimensional miniature organs that capture major functions, structure, and the histological characteristics of the organ that they're modeling. There has been thriving research related to organoids around the entire globe, and as a result of that, there has been different ways to grow these organoids. It is possible to grow organoids from embryonic stem cells or induced pluripotent stem cells, and another way to grow stem cells is using adult biopsy tissues from the regenerative organs. These organs could be lung, stomach, liver, and intestine, for example, that have stem cells in them, which can give rise to these organoids. At ACROBiosystems, we hope to be of some help by focusing on iPSC-derived organoids to complement the other types of organoids. So let's look at the evolution of organoid technology over time. About 120 to 60 years ago, researchers discovered the regenerative potential of some organs, or in some cases, some animals. So they were dissociating some animal organs or dissociating some animals like sponges, and they were seeing that these dissociated cells can reaggregate themselves. And about 40 years ago, the emergence of stem cell biology helped us to understand that these differentiated cells can organize themselves into structures similar to how they're found in tissues. And about 20 years ago, the iPSC technology was invented by Yamanaka's group, and in this technology, fibroblasts are genetically reprogrammed to regain their pluripotency, and the induced pluripotent stem cell would have the capacity to differentiate into almost any type of cell in our body. And about 15 years ago, the first tissue stem cell derived organoid was developed by Clever's group. In the last 10 years, different types of organoids were gradually developed. And this you can also see reflecting into the number of published papers about organoids overall, or more specifically, iPSC-derived organoids. This shows us that the base technology of organoids is now mature enough to support the large-scale commercialization of organoids. So let's look at the current commercialization of organoid, where it is now. In the last 10 years, we do see the total funds increasing, raised by organoid companies. And if you look at the organoid market size, it is predicted that it raises up to $12.8 billion by the year 2030. So there is an increasing number of pharma, reagent companies, CROs, and institutes that are adapting this technology. And the commercial growth is increasing. Another reason to adapt organoids into drug discovery is the regulatory agencies' allowances. So several regulatory agencies, such as FDA in 3 continents, currently allow the usage of non-animal models in the drug discovery phases. And there's also the pharmaceutical companies, the top pharmaceutical companies supporting the organoid technology, as you can see here. So let's look at the pain points of global drug development. We know that even after rigorous preclinical testing, when switched to the clinical stages, almost 90% of the drugs fail due to effectiveness issues or safety toxicology issues. This can be attributed to the preclinical models that are currently being used, such as the 2-dimensional cell lines that lack the physiological representation, or the rodent models that lack the human similarity, the genomic and physiological similarity, or the non-human primate models, which are costly and bear ethical issues. So it is now a good time to adapt the organoids as an alternative to these models that have these problems to combat high clinical drug failure rates. What about the iPSC-derived organoids? So there are some special advantages to using these kind of organoids. Firstly, with iPSC-derived organoids, you would have a certain level of genomic and transcriptomic stability between batches if you use the same donor iPSC. Therefore, this allows you to have comparative results when you test the efficacy or toxicity of your drug. And this allows you to have a large production, so you would be able to screen your drugs in a large scale, in a high throughput manner. And comparatively speaking, although the maturity of iPSC-derived organoids is not as high as that of the adult tissue-derived organoids, transcriptomically or epigenetically, the complexity that you would reach with iPSC-derived organoids is much higher. So you would have more similar cell types to that tissue. And iPSC-derived organoids give you the possibility to grow organoids from non-regenerable organs, such as the brain, that have very low amount of stem cells. So this gives you the unique advantage to grow organoids from these kind of tissues. And obviously, you have a human genomic background, so the response created from this genomic background would be quite similar to in the clinical studies, and they bear no ethical limitations. And here there is a lot of human iPSC-derived organoid disease models developed. By the year of 2023, approximately 35 human iPSC-derived organoid disease models in all of these organs and in all of these disease models were created, which shows that it's expanding, and there is a lot of research accumulated currently on these disease models. Looking at the economic perspective, we have compiled from the Clinical Intelligence Sideline website what are the current preclinical pipelines ongoing. So we saw that they focused in certain organ models such as liver, heart, CNS, and intestine. And in each of these organ models, for example, for CNS diseases, Alzheimer's, Parkinson's, ALS, and pain were the top categories. For liver diseases, non-alcoholic steatohepatitis, hepatoma, dysfunction, fibrosis, and for cardiovascular disease, it was ischemia, heart failure, hyperlipidemia, hypertension. And at ACROBiosystems, we focus our efforts to model these types of diseases. So you would find heart failure toxicity models, Alzheimer's disease, Parkinson's disease, NASH, non-alcoholic steatohepatitis, inflammatory bowel disease models that we model in our organoids. So it would be a good time to ask, how realistic are these organoids? How realistic is the resemblance functionally, structurally? It's good to remember that they are not exact replicas of the organ. For example, a brain organoid does not get meaningful input as our brains do, or does not give a meaningful output as our brains do. However, this technology allows us to gain insight of the hidden biology of these complex organs that we never had the chance to have so far with the current models, such as the animal organs, two-dimensional cell cultures, and human autopsy tissues. And how well it works is quite surprising. So I will now walk you through the different ACROBiosystem organoid solutions. At ACROBiosystems, we leverage our strength in delivering high-quality proteins to nurture these complicated three-dimensional cell models. And in each of these organoid types, we have vascular components, which is comprised of an endothelial cell network, an immune component, and these come as ready-to-use organoids, so they are shipped live to you. And also the differentiation kits for these are available. So we would focus on heart, brain, intestine, and liver models. And here you can see the types of disease models we will show you in the next slides, from heart toxicity to Alzheimer's, Parkinson's, IBD, and NASH. Let's start with the cardiac organoids. So these organoids are quite convenient solutions. These cardiac organoids can be obtained only in 2 weeks. They have a rich endothelial cell network, and they have mechanical beating that can be seen by naked eye even, and they have heart chambers that can also be seen by naked eye. Generally speaking, cardiotoxicity is mainly centered around a few points. This could be a direct cell damage to one of the cell types of heart, like cardiomyocytes or coronary artery or fibroblast degeneration, or it could be acting to the ion channels, resulting in rapid changes of these ions that would reflect to the heart beating behavior. So these cardiac organoids, they have multiple chambers similar to the heart chambers. And there is even atrial to ventricular specification, atrial to ventricular compartmentalization, which you can see in green, in red. And these organoids also have an endothelial network, as you can see, marked by CD31 marker. Therefore, this gives you the possibility to evaluate your drug either directly at the atrial cardiomyocytes or ventricular cardiomyocytes, more specifically, or to the endothelial cell network. Sorry, my screen froze. Okay. So we also validated the functionality of these cardiac organoids when they're treated with a drug, a cardiotoxic drug. So we looked at how the cardiomyocytes and the endothelial cells were affected in response to that cardiotoxic drug. As you can see, when they're treated with the drug, the endothelial cells as well as the cardiomyocytes were both damaged. And we also used a drug that causes the prolongation of the QT interval. And we measured the response of these organoids with a multi-electrode assay. And as you can see in the red line, this drug functionally also caused the cardiac organoids to have a prolonged QT interval. Of organoid model is cerebral organoid. So these cerebral organoids, we have developed this protocol as well to allow modeling different brain diseases. And this is also a quite simple protocol. These organoids have IBA1 positive cell population, which is the immune component, a vascular component, and dopaminergic neurons expressed by tyrosine hydroxylase positive markers. So when these organoids mature, the neural rosettes would then differentiate into more mature neurons, and then the brain would have a more mature network. And here you can see when they're produced in size, they show a very nice morphological consistency when grown. So these organoids also inherently show several cell types that you would find in the brain. For example, endothelial cells, neuronal progenitors in the earlier phases. And then you would also see mature neurons, astrocytes, oligodendrocytes, dopaminergic neurons, microglia. So in the later phases, you start seeing microglia markers, and dopaminergic neurons allow you it's crucial for modeling Parkinson's disease modeling. Therefore, we also have these kind of cells. And we also functionally validated the electrophysiological activity, the electrical activity of these cerebral organoids by several assays. So we have performed MEA analysis, silicon probe analysis, where we stick the silicon probes in the core of the organoid to measure the activity there, as well as patch clamp assay. And we do see a spontaneous electrophysiological signal overall, validated by all these three models. So in the past, both Alzheimer's and Parkinson's disease models focused heavily on animal models. And it is also possible technically to model this in larger animals. However, this brings like another set of difficulties. Here in ACRO, we have focused our efforts to model these two diseases using our cerebral organoids. So we do this by using preformed fibrils, which are polymerized proteins, in simpler terms, that allow you to model these diseases in a simple way. And another way is to use chemicals. As you know, protein aggregation is one of the key hallmarks of diseases like Alzheimer's and Parkinson's disease. When you add these preformed fibrils made up of either tau or from alpha-synuclein, they act as seed structures, and it recruits even more proteins. And this triggers the cells to show Alzheimer-like or Parkinson-like phenotype. So this allows you to model these kind of diseasing, which is a non-genetic, non-chemical, non-physical way, a novel way to model these types of diseases that is better mimicking the naturally occurring pathological states of these processes. And these preformed fibrils are also standalone available on our website. When we use tau preformed fibrils, this allows us to create Alzheimer's disease models simply by the addition of these preformed fibrils to the organoid culture solution. So the cell medium, basically, when they are treated with the preformed fibrils in comparison to the negative control, you can see that the phosphorylation pattern of tau is increased in a dose-responsive way even. So therefore, you do see the hyperphosphorylation hallmark of Alzheimer's disease when they are treated with tau preformed fibrils. So this is one way to model Alzheimer's disease in a simple and non-genetic, non-chemical way. We also used another molecule called afton-4. So this is a small molecule that induces hyperproduction, hypersecretion of amyloid beta 42 peptide when added to cerebral organoids. And this can be used to model Alzheimer's disease as well using the cerebral organoids. So afton-4, when added to these organoids, when they're treated with afton-4, as you can see in day 3, they start showing degenerative behavior. They start degenerating, and even in the media, you start seeing a lot of debris. You start seeing in the afton-treated organoids on the tissue culture plate a crystal-like aggregate upon treating with this drug. And we're currently analyzing these aggregates, whether they are amyloid beta 42, knowing that this small drug, is inducing hyperproduction of this peptide. And we also looked at the dendritic structure, dendritic network of these cerebral organoids when they're treated with this drug. So MAP2 is a dendrite marker, and when the organoids are treated with afton-4, you can see the dendritic network to degenerate quite a lot. And here you can see a lot of cell bodies. However, very few dendritic structures caused by afton-4 induction, which is another way of modeling Alzheimer's disease. These preformed fibrils can also be used to model Parkinson's disease. So in the case that alpha-synuclein preformed fibrils are used, which look like this under electron microscope, it is a good strategy to see the effects on the dopaminergic neurons or the tyrosine hydroxylase positive neurons more specifically. So in red, you see the tyrosine hydroxylase positive neurons. And when they're treated with alpha-syn preformed fibril in lower and higher concentrations, you see that the red network to be degenerated quite highly. Not only the tyrosine hydroxylase positive dopaminergic neurons are affected here, you also see abnormal network effect of all of the other dendrites that are marked by MAP2 staining here. So you see an overall degeneration caused by the alpha-synuclein preformed fibrils, which is another way to model Parkinson's disease. We also utilized chemical methods to model Parkinson's disease. So 6-hydroxydopamine is a chemical when added to midbrain. The animal shows Parkinson's-like symptoms. Chemical is added to organoids. In control, you see no degenerative effects. However, when this chemical is added, after only 24 hours, you start seeing a degenerated organoid. And we also tried to rescue this effect by using a protectant, and this protectant is 2 growth factors and a vitamin. And we were able to see that when this protectant is added, even in the presence of this chemical, there was higher cell viability, no morphological degeneration response. And we also wanted to see the cell-specific effects of the 6-hydroxydopamine, as well as when it is tried to rescue with the protectant. So when the 6-hydroxydopamine is added, you can see that the dopaminergic neurons are degenerated. And in the addition of, in the case that the protectant is added, you can see the dopaminergic neuron network being preserved. Therefore, also in a cell-specific way, we were able to model and rescue this disease type in our cerebral organoids. So the next type of organoid I'd like to tell you today is intestinal organoids. They have various cell types. They are able to mechanically contract these peristaltic movements, the intestinal contractions, and they are also highly homogeneous. And their unique feature is that they are able to grow up to a diameter of 2.5 centimeters with no viability issues in the core. So they're quite large. You can see an intestinal organoid at day 75. When it is cut and unfolded, you can see under the microscope that there are certain ridges similar to Pelisae circularis. If you look at the organoids here, you can see that these are quite large, and in comparison to a cone, these can be grown up to very large diameters. Histologically, there are several cell types present, and there are lumens here similar to intestinal lumens. So like the inside of the intestine that can be seen by whole organoid sections. So you can see intestinal crypt-like, smooth muscle-like, adipocyte-like, and granulocyte-like histology when looked at in detail. And we also validated the presence of smooth muscle cells and epithelial, macrophage, goblet, stem cells, enteroendocrine, and endothelial cells by immunofluorescent stainings. And we also validated the cell types available with RNA sequencing, and you do see a proof of the existence of these cells in the RNA level as well. We also functionally validated these intestinal organoids because you would expect the intestinal organoid to absorb nutrients. When fatty acids or glucose is given to these organoids, you can see that they are able to absorb these nutrients. Therefore, this is also another proof of a functional of the functionality of these organoids. To create inflammatory bowel diseases from these intestinal organoids, there are several methods to do that, and we have used the LPS stimulation method. So when these intestinal organoids are stimulated with LPS, due to the immune reaction that are caused by LPS, you start seeing an IL-6 and CCL2 expression to increase upon LPS stimulation with higher concentrations. This shows that there is this active immune components, and this immunized intestinal organoids serve as an IBD model. The next type of organoids I will mention today is the liver organoids, so the vascularized liver organoids. They also have a simplified protocol. The hepatocytes are available, bile ducts and vascular cells are available in this model. And you even see liver lobule-like structures. Liver lobules are small units found in the liver, in the human liver, and you start seeing similar structures in these organoids as well. And we also have looked at the critical enzymes produced by liver, such as albumin, CYP50 enzymes, and BSEP. So we wanted to compare the level of these enzymes to immortalized cells, such as the HepG2 immortalized liver cell. You can see that our human liver organoids are able to produce these enzymes in a higher level. It is difficult to reach the primary human hepatocyte levels. However, our ACROorganoids were able to produce these enzymes in a higher level compared to the immortalized cells. We also looked at the effects of a liver-harming antibiotic. When this antibiotic was used on these organoids, one of the critical enzymes was increased in response to this organoid. So they were functionally responding to that liver-harming enzyme, to that liver-harming drug. To create NASH models, liver non-alcoholic steatohepatitis model, we have utilized the oleic acid and LPS stimulation. One of the hallmarks of NASH is induced lipid drop accumulation and increased inflammation. When these organoids are treated with oleic acid in lower and higher concentrations, you start seeing more production of the lipid droplets. And this is even more rigorous, more of a drastic, you start seeing a more drastic effect when they are cultured for longer times, especially for the higher concentration. So you can see that in the hepatocytes seen by low light transmittance, there is increased lipid droplet accumulation caused by oleic acid treatment. And we further immunized these organoids after the addition of oleic acid by using LPS. And you can see that when LPS is used, an immune reaction was created. So there were increased levels of IL-6, TNF-alpha, IL-10, as a response to LPS stimulation. So this serves as a NASH model, having more immune reaction, so being immunized and showing more induced lipid droplet accumulation as an organoid NASH model. And you can even see these lipid droplets in white, shown by the arrows, and this serves as a liver organoid NASH model. So with that, we come to the end of these disease models, and I'd like to mention some future perspectives of these organoids. So as you have seen so far, in these organoids, there are several relevant cell types, an immune component, vascular components, histological, structural, and functional resemblance. And they represent as excellent models to respond to drugs in a similar way as an alternative method, as an alternative preclinical model. Therefore, this shows that these are precise human in vitro disease models that can be used for both research and for drug development purposes. And we think that these translatable results with using human iPSC-derived organoids will fast-track the preclinical stages, showing more accurate response of the efficacy and the toxicity caused by these drugs. So with that, I'd like to thank you all for your time. Thank you for listening. Please don't hesitate to reach out to me or to our team for any inquiries. And I would be happy to answer your questions now. So thank you very much.
Just like Idil said, we're just going to now move on to the Q&A session. Sorry, I can hear some feedback. Apologies about that. I can see quite a few, actually a couple of questions from the audience. So let's start with that, if that's okay with you, Idil. So one question here is from Catherine, and it says, hello, how do you ship the organoids to the customers? Are they frozen or alive? If they're frozen, how did you freeze them to ensure proper survival and functionality after thawing? How long does the shipment take? Do you have several sites from where you ship them to customers worldwide?
Thank you very much, Mary, for reading the question, and thanks for the question. So we do ship the organoids live currently, and soon we will also provide the cryopreserved ones. So currently they're shipped live in a hibernation medium and in thermostable containers. We do ship only from our R&D facility, which is located in Beijing. Therefore, we are currently expanding our organoid facilities to the US side as well. However, currently we only ship from the Beijing. And the shipment takes about 1 to 2 weeks, which we validated to have good survival rate. Once they arrive, they are transferred to a recovery medium. And for the cryopreserved ones, we developed a special formulation to have good viability after cryopreservation. So I hope this answers your question.
Thank you, Idil. I hope so too. But Catherine, please do let us know if not, and please do get in touch with us, and we can set up a meeting or discuss further. Another question from the audience, Idil, is: do you give the glucose and fatty acid in the media surrounding the intestinal organoids, or do you inject it in the middle of it? The absorbing part would be inside of the organoid, right? And that's a question.
So we did put them into the media, but it is also possible to inject them into the inside lumen. So they're absorbed from the outside and from the inside as well. So technically, it is possible to inject them to see how it specifically absorbs it from the inside.
Thank you very much, Idil. Once again, I hope this answers the question. But if not, please do let us know or do get in touch. And yeah, hopefully we'll be able to help. We have another question from the audience. Nice talk. With the increasing interest in RNA therapeutics, could you share more on how organoids may be used to study delivery of RNA drugs? Example, will the matrigel prevent delivery of RNA probes?
So one unique advantage of our organoids is that we don't have matrigel embedding. So we don't need to embed these organoids into matrigel, which allows certain physiological recordings, functional recordings, as well as for RNA therapeutics possibly. They kind of grow their own extracellular matrix because like in the earlier phases, this takes a few more days to let them produce their own extracellular matrix, but then this comes with the advantages of having a more physiologically relevant ECM. So they can be used to test the efficacy of these RNA drugs overall. So using any type of model that you're targeting, it would be possible to screen the efficacy of these RNA drugs, definitely.
Thank you very much, Idil. I think that was a great answer. Once again, I hope everything was clear. If not, please get in touch. We also have another question from Allah, from the audience. They say, hello, thank you for this presentation. What do you think the regulatory classification would be for these organoids?
So the regulatory classification, they need to be cultured in S2 labs, so safety level 2 labs, and there are no other regulatory issues with that. there might be like some country-specific regulatory issues due to iPSCs, but overall there is no ethical, like you don't need to write like an ethical allowance or so, similar to how you would do for animals.
Perfect. Thank you, Idil. Sorry for all the questions. Just a couple more.
I'm happy to answer them.
Perfect. Thank you very much. So we have one more question from the audience. They ask, how can you grow hydrogel-free organoids?
So I just briefly answered this. So to go a bit more deeper, we use like self-made formulations, relatively high growth factors and so on to grow these, to trigger differentiation of these cells. These cells, they also, like for example, during the development of human, the cells also form their own extracellular matrix, right? Therefore, we follow a similar strategy and we let the cells produce their own extracellular matrix. Therefore, in that way, they are able to stay as a three-dimensional spherical organoid model.
Thank you very much, Idil, for that. I just realized I didn't mute myself. I hope there wasn't no feedback. A few more questions. So we have a question from the audience as well that says, hello, thanks for the comprehensive talk. What are the limitations of organoid lab work?
So the limitations depends on your previous experience with organoids. So it could be relatively, for some organoid models, it could be relatively longer maturation times of organoids, or it could be having no experience with cell culturing. So our protocols are relatively easier to grow. So you just have media formulations, you mix them and you treat the cells. Well, it's a must to start with good quality iPS cells. However, it's good to, so using these protocols that we provide you, it would be relatively easy. And if not, we would provide with consultancy. So we would help you to troubleshoot. So this would kind of be a way to overcome these limitations. Other than that, it's relatively easier. And if you would have problems with time, so no allocated like resources to, or no allocated employees or students to grow these organoids, it is also possible to ship them ready to use. So we would just ship you the live ones so that you don't have to wait and you don't have to grow these yourself. Other than that, we are trying to standardize this protocol and simplify this protocol so that it is available to everyone who is willing to set up an organoid facility or who's willing to use organoids in their research or drug discovery purposes.
Thank you very much, Idil. One more question. How does the vascular component look like in organoids? Are there functional vascular tubes?
These are not exactly vascular tubes. So the vascular component is comprised of endothelial cells. These endothelial cells form a network in between, so they kind of interact and basically show a network structure comprised of endothelial cells. However, they don't form functional blood vessels, functional tubes with blood, so there's no blood flow or anything. However, this gives you the opportunity to monitor the effect on the endothelial cells, for example. So currently, this is how the vascular component is.
Thank you very much, Idil. Again, if something is not particularly clear, please do get in touch. We can have a more comprehensive meeting and discuss questions such as this in more depth. Some more questions. Do you also provide services such as toxicology, efficacy testing on organoids, etc., if we don't prefer to order organoids but have the testing done in your facilities? Great presentation and thank you.
Could you repeat the question? I think I missed where it is.
Yeah, no, sure. So the question is, do you also provide services such as toxicology, efficacy testing on organoids, etc., if we don't prefer to order organoids but have the test done in your facilities?
Yes, so we also offer services of toxicology or efficacy testing in our facilities, in our R&D facilities. So if you don't want to touch any of the work, it is possible to ship us the drugs that you would want to test or the other chemicals that you want to test, and we would take this as a service offer, as a service project. So it is possible. Thank you very much for saying a great presentation.
Perfect. One more question from Catherine. Do you have quality certificates for the organoids? Are they meant for R&D only?
As far as I know, they are meant for R&D only. Yes.
Perfect. And Catherine again is asking, how long are they supposed to be functional after receiving them?
It depends on the organoid type. So they can be cultured for, in some cases, months. For example, for cerebral, it's even much longer. I guess the question is, how soon can I use them after shipping? So firstly, for about 2 days, you need to put them, like transfer them in a recovery medium with a lot of growth factors, with high concentration of growth factors, and then they can be used. But for example, for cerebral ones, you would want to see what your aim is. For cerebral ones, you may want to mature them for another month or so. But technically, they are functional organoids after about 2 to 3 days after receiving them. So they will be ready to use.
Thank you very much, Idil. And I see we have one last question. I don't see much on lung organoids. What are your thoughts on them in regards to developing one? If you do have lung organoids, are they being developed without Metrogel as well?
Yes, so we are currently also developing our protocol to grow lung organoids as well. So soon we will also offer that, and most likely they will also be grown without Metrogel as well.
Thank you very much. I believe those are, we have one comment in the chat from Shay. Sorry if I mispronounced your name. They say, thanks for the clarification. Next time we would like to know other organoids, for instance, lung organoid information. Thank you again.
Yes, so let's stay in contact, and we would be happy to provide you with the updates of the lung organoids. And please reach out to us if you have any specific inquiries, if any specific wishes.
There's also been one last question being put through the chat. Anonymous is asking, I saw that you also have differentiation kits to grow these organoids. Can we grow them in our facilities?
Yes, so each of these products, we can ship these organoids live to you, but in addition, we also offer the differentiation kits that we use, that we developed ourselves, to grow these organoids from the iPSCs in your facilities as well. So if you have an organoid facility, it will be possible to use the differentiation kits and grow these organoids themselves. If you don't have an organoid facility, we could ship these organoids to you live, or we also offer services to screen drugs for efficacy and toxicity in our facilities as well. So all these types of offerings are available from ACROBiosystems.
Perfect. I do believe now that that's the end of all the questions. Once again, thank you everybody so much for joining us today. The end of the Q&A session marks also the end of our webinar. Please follow us on LinkedIn, scan the QR code that's on your screen right now. Please follow us on LinkedIn so that you stay updated regarding the rest of the webinars in our series. And we hope to see you again soon, and we hope you also enjoyed yourselves today. Bye-bye, everybody.
Thank you. Bye.